Good morning. Today we are here to discuss about nervous tissue, which is one of the fundamental basic tissues in the human body. Among the four tissues that we have uh, seen, nervous tissue is the fourth type, which is one of the most elementary and I will say basic uh, functional unit which kind of integrates all the different tissues to a functional mode, which is regulating the functions in the body to a great extent. So, let me start off with what it is made of. So, nervous tissue is basically made of two structures, neurons and neuroglia. So the functional unit is neurons, and the supporting cells or the connective tissue which supports the neurons we can call as neuroglia. So among the different types of neurons and what are some of the most important neuroglia we shall see in this discussion. So before that, about the basic classification of how we can classify the different types of nerves. Now, nervous system we can broadly divide into central nervous system, a peripheral nervous system, and an autonomic nervous system. So the central nervous system is divided into the brain and the spinal cord. The peripheral nervous system is divided into the peripheral nerves, which arise from the central nervous system, and of course the groups of neurons that may be seen along with it, which we call as ganglia. And then there is this third type called the autonomic nervous system, which is actually uh, together with the central and peripheral nervous system. But when it regard to its function, it is divided as two types. It is parasympathetic or and sympathetic type. So these are the basic classification of the nervous system and its components. So neuron is the functional unit, as I mentioned, and it is made of a cell body which has got particular distinct feature from all the other three types, which is we can which should be mentioned here. That is the Nissl granules, which makes the neuron cell body quite distinct and which is a modified type of rough endoplasmic reticulum. And then, of course, the processes that is arising or attached to the cell body. So there are, according to the types of processes, of course, there are again other classifications for neurons, but this is the most commonest type of neuron that we might see, uh, called as multipolar. In this case, we can see in this diagram that there are several short processes and a long process. So this is what generally, when a neuron diagram is being shown, but this is one type of neuron which we call as multipolar, which means there are several processes coming from the cell body. So the processes, as I mentioned, can be the short ones, which we call as dendrites, and a long one, which is called as axon. So generally, a neuron will have one axon only, whereas the number of dendrites can depend upon the type of neuron. Okay, so this neuron or these axon processes could be covered by a protein which we call as a myelin sheath, or as well as there are axons which are not covered by this myelin, so they will be called as unmyelinated axons, whereas those which are covered by this myelin is called as a myelinated axon. So this is the myelin sheath, we call it, that covering is called as a myelin sheath. It is a process which is arising as a result of Schwann cells in the peripheral nervous system. This is what we see as a Schwann cell, and you can see the axon which is invaginating into it, and then this arise this axons or the myelin around it is being covering, as you can see, it is coiled up, and several such coils together make up a myelin sheath. So such axons are called as myelinated axons. In the case of the peripheral nervous system, we see this more. And in the central nervous system, the myelin sheath is not provided by Schwann cell, but it is provided by another neuroglia called as oligodendrocyte. So this is just to mention the what is a myelinate. Now, when, as I said, depending on the type of processes, we can classify neurons as multipolar, which I already showed. And then you have bipolar, which means there is only two processes, that is one axon and a dendrite. And then you have unipolar, which is normally as pseudo-unipolar. The pseudo-unipolar will have one process, and functionally we may see that it is classifying into, or it is diverging into branches. So these are the type of neurons. Now, the connective tissue support for neurons, that is neuroglial cells. Neuroglial cells can be of several types. In the central nervous system, we have ependymal cells, which are otherwise epithelial, modified epithelial cells, which line the cavities called ventricles. Then, as I just mentioned, oligodendrocyte, which is providing myelin sheath for the axons in the central nervous system. Then astrocytes, which are some star-shaped microglia. These are all seen in the central nervous system as supporting nerve cells. And in the peripheral nervous system, we have satellite cell, Schwann cell that we just saw. See, these are just diagrams showing those. I can briefly mention some important aspect of some of these cells. That is, astrocyte is star-shaped, as the name suggests. It is a very important structure which helps to form the blood-brain barrier. And then we have microglial cells, which is very much related to the macrophage activities. So it is important for immune system, or helping to keep the nervous system intact and maintaining it to a very great extent. Ependymal cells are those specified modified cells which are seen in the cavities of the brain, which we call as ventricles of brain. So these are, then you have oligodendrocytes. Oligodendrocytes are specialized cells, which I told, which provide myelin sheath in for the neurons or the axon that we see in the central nervous system, whereas in the peripheral nervous system, the myelin sheath is provided by the Schwann cells. Then you have satellite cells, which may be seen more in the ganglia in the peripheral nervous system again. So these are some of the neuroglial cells. Now, a very important structure wise in the nervous system is this classification called gray matter and white matter. So what is gray matter? Gray matter is where you have collections of cell bodies with its short processes, and also where you may see unmyelinated axons. Such groups are such uh, collection is called as gray matter, whereas white matter is full of bundles of fibers, which normally are myelinated axons. So because of the light reflection in these myelinated sheets is why it appears as a little more lighter or white in appearance during the histological stainings. So that is what is generally classified as gray matter and white matter in a very brief term. Then the same gray matter, if they are dispersed within this central nervous system, that is either inside the white matter or isolated away from the continuous gray matter, that we call as nuclei. So small bodies of gray matter collections or cell body collections, they are called as nuclei with regard to the central nervous system. And the same kind of aggregation, if it is seen outside the central nervous system, we call them as ganglia. Is a plural term, ganglia. Ganglion is otherwise the singular term. And ganglia is where you see the cell bodies of neurons in the peripheral nervous system, mostly connected to a spinal nerve um, or its branch. So these are some of the uh, types of most familiar terms and structures that we will see with the nervous tissue. Now, what is white matter? White matter is, as I told earlier, it is collections or bundles of axon fibers. Or a nerve is a collection or a protoplasmic processes. So a collection or a bundle of protoplasmic processes of axons. So when such nerves or such fibers are aggregated, you call it as a tract. And these are seen basically in the spinal cord, either traveling up or traveling down. So we call them ascending or descending tracks. And several such tracks, when they collect together, you get a little more bigger bundle, and that we call as funiculi or fasciculi or lemniscus. This we can see as we progress up the spinal cord towards the brainstem. And then furthermore, when you see bigger collections, or we can say the largest collection of fibers, we can call them as peduncles, which may be connecting it to the particular parts of brain like cerebellum or to the cerebrum. So we have cerebral peduncles, we have cerebellar peduncles. So these are much more bigger bundles. So this is how the white matter is organized in the central nervous system in a very brief manner. So, coming to peripheral nerves, these are what are attached to the central nervous system. There are two types. One is the spinal nerve, other is the cranial nerves. So, spinal nerve is what is are those nerves which are coming or arising from the spinal cord. There are 31 pairs of spinal nerves which are arising from the spinal cord through the intervertebral foramen of these vertebrae that we protects the spinal cord. So that is, those are called as the spinal nerves. Whereas there are also nerves or peripheral nerves which arise directly out of the brain and brainstem. Such nerves are called as cranial nerves, which we have 12 pairs. So 12 pairs of cranial nerves which arise from the brain and or the brain stem and the brainstem. That is also peripheral nerves. So these peripheral nerves which arise from the brain directly are called as cranial nerves, and they are mostly innervating the head and neck structures, and also some of the viscera organ in viscera in the thorax, in the abdomen, and so these may be coming from the cranial nerves. So peripheral nerves which arise from the spinal cord are spinal nerves, and peripheral nerves which arise directly from the brain are called as cranial nerves. So this is how the peripheral nerve is just organized in its distribution. Formation of a spinal nerve will involve both sensory fibers as well as motor fibers. So a spinal nerve uh, is a mixed nerve with regard to the fibers that or the function that it is carrying. So it will carry sensory impulses as well as motor impulses. Whereas cranial nerves, not all cranial nerves are mixed. Some of the cranial nerves are purely sensory only, and some are purely motor only. So like olfactory nerve, optic nerve, these are purely sensory nerves, whereas there are few which are like oculomotor nerve, trochlear nerve. These are purely carrying only motor impulses. Well, while there are a few cranial nerves which are carrying both motor as well as sensory, like the trigeminal nerve, the facial nerve, the vagus nerve. These are glossopharyngeal. These are mixed nerves which carry both sensory and uh, motor fibers. So in that way, cranial nerves are a little different from how the spinal nerves are organized, but or function. So histologically, these peripheral nerves, we can see they are having several coverings which protect these fibers. One is the innermost covering of one axon, of course, over the neurilemmal sheath is what we call as the endoneurium. And then several such endoneuriums which are bundled together are protected by a connective tissue covering which we call as perineurium. So several uh, perineuriums, or we can call them as nerve bundles. So several nerve bundles which are covered by this perineurium. When they are bound together by a bigger connective tissue covering, that is epineurium, that forms a nerve, or that is what we call as a nerve. So a bundle of epineurium, which is of course covering several bundles of perineuriums, within which within a perineurium where you can see several small endoneuriums which are protecting each axons or fibers. So this is how the organization of the peripheral nerve is with regard to how it is structured. So as a highlight to review what we have seen so far, it is we have the several um, types of cells within a nervous tissue, which we broadly classify as just two types, that is the neuron and then the supporting cells which are called as neuroglia. So neurons are the fundamental unit which has got a cell body and process, either a singular process or double process or multipolar processes. So depending on that is one classification of neurons, and the connective tissue that supports them, which we called as neuroglia. These are supporting cells like astrocytes, oligodendrocytes, Schwann cells in the case, and microglia. So these are all connective tissue neuroglial cells. And depending on how they are organized, we can see we saw there was gray matter and white matter. Gray matter is where we had collections or aggregations of neuronal cell bodies with unmyelinated axons, is called as gray matter. And white matter is where we see predominantly axons or myelinated axons together. We call them as white matter. And then we saw there could be small dispersed masses of gray matter within the central nervous system, which we called as nuclei. And then there was the same, if it is seen outside the central nervous system, we call as ganglia. And then basic classification when it came to the peripheral nerves, we saw there are those nerves which arise directly from the spinal cord, which we call as spinal nerves, and those which arise directly from the brain, which we call as cranial nerves. So this is just the basic structure of how the nervous tissue elements are seen within our nervous system. So I hope it is clear to us regarding the basic structure of the nervous tissue. Thank you.